Hi friends, in the last video we discussed about DNA vaccines. In this video we are going to discuss about synthetic peptide vaccines and multivalent subunit vaccines. Synthetic peptide vaccines. They are usually composed of 20 to 30 amino acids and they contain specific epitope of an antigen related to that specific infectious or chronic disease, which may also include cancers. These vaccines consist of synthetic peptides, carbohydrates, or antigens. They are usually considered safer than vaccines from bacterial cultures. Since these vaccines are prepared synthetically, their production can be faster. Synthetic peptide vaccines represent fragments of protein antigen sequences. Therefore, they synthesize specific B cell and T cell epitopes, which offer the potential to induce the disease's neutralizing immunoresponse with completely synthetic structure. The short chain peptides can be used to mimic antigenic sites of viruses and can be used as the basics for vaccines and development. The advantages: their antigens are precisely defined, and also the side effect causing unnecessary components are avoided. They are stable and their production and quality control is simpler. They are also less toxic as they do not contain viral or external proteins. They are safer in cases where viruses are oncogenic or establish a persistent infection. Disadvantages: this includes they are less immunogenic than conventional inactivated whole virus vaccines. They require adjuvant and also primary course of injections, which is followed by boosters. They also fails to elicit CMI, otherwise cellular mediated immunoresponses. Next is multivalent vaccine. The synthetic peptide vaccines and recombinant protein vaccines have the disadvantage that they tend to be poorly immunogenic and do not induce cell mediated response. These are their main disadvantage or their limitation. So there comes the multivalent vaccine. The multivalent vaccines present multiple copies of a given peptide or a mixture of peptides to the immune system. And they cause both humoral and cellular mediated immune responses. So, in this multivalent vaccines, there are two approaches where we can prepare this vaccine. So, the one of the approach is to prepare SMA or solid matrix antibody antigen complexes. These complexes are made by attaching the monoclonal antibodies to a particular solid matrices and saturating the antibody with the desired antigen. The resulting complexes and then can be used as vaccines. By attaching different monoclonal antibodies to the solid matrix, it is also possible to bind a mixture of peptides or proteins composing immunodominant epitopes for both T cell and B cell to the solid matrix. These multivalent complexes have been shown to induce vigorous humoral and cell mediated responses. Their particular nature contributes to their increased immunogenicity by facilitating phagocytosis by phagocytic cells. So here we can see the figure of one of the approach of creating a SMA complex. So here we can see the solid matrix antigen antibody complex is designed to contain synthetic peptides representing both T cell and B cell epitopes. And thus the SMA complex is formed here. And Another approach is by means of producing a multivalent vaccine using detergent to incorporate protein antigens into protein micelles or liposomes, lipid vesicles, or immunostimulating complexes. Immunostimulating complexes are also known as ISCOMs. Okay, and the mixing proteins in detergent and then removing the detergent forms the micelles. The individual proteins orient themselves with their hydrophilic residues toward the aqueous environment and the hydrophobic residues to the center as to exclude their interaction with the aqueous environment. Liposomes contains protein antigens which are prepared by mixing proteins with a suspension of phospholipids under conditions that form vesicles bounded by a bilayer. The proteins are incorporated into the bilayer with the hydrophilic residues exposed. Immunostimulating complexes are lipid carriers prepared by mixing protein with detergent and a glycoside called quil A. So here we are forming three types that are liposome, micelle and immunostimulating complexes. So we can see how they are formed. A pictorial representation. Here we can see that detergent extracted membrane antigens or antigenic peptides. Here we can see that immunostimulating complex is formed by using a protein mixed with a detergent and a quil A. And here we can see the formation of liposome as we told earlier, and also the formation of micelle. Protein micelles, liposomes and immunostimulating complex can all be prepared with extracted antigens or antigenic peptides. In micelles and liposome, the hydrophilic residues of the antigen molecules are oriented outward. In immunostimulating complexes, the long fatty acid tails of the external detergent layer are adjacent to the hydrophobic residues of the centrally located antigen molecules. So, here we can see the centrally located antigen molecules. This picture shows how an immunostimulating complex is delivered into a cell, or the antigen inside the immunostimulating complex is delivered into the cell. Here we can see that they deliver the antigens inside the cells and they mimic the endogenous antigens. The subsequent processing by cytosolic pathway and presentation with the class 1 MHC molecules induces a cell-mediated response. The membrane proteins from various pathogens, including influenza virus, measles virus, hepatitis B virus, and HIV, have been incorporated into micelles, liposomes, and immunostimulating complexes, and are currently being assessed as potential vaccines. In addition to their increased immunogenicity, liposomes and immunostimulating complexes appear to fuse with the plasma membrane to deliver the antigen intracellularly, where it will be processed by the cytosolic pathway and thus induce a cell-mediated response. This we saw in the last figure of immunostimulating complex, how they give the antigen to the cell. Thank you.